Good morning, everyone. Good morning, and as people are taking their seats, I'm just hoping that everybody had a lovely evening, found some wonderful food. It's your own fault if you can't find it here in Montreal. It's a wonderful food experience. Again, we're blessed with another gorgeous day out there, and I hope you get some chance to see the sunshine. We're very excited this morning to have our first speaker for of the day be this basic science lectureship, which is sponsored by McKeith Press, publishers of the Academy's official journal, Developmental Medicine and Child Neurology. Professor Bernard Dan, the editor, will be introducing the lectureship. Thank you.
Thank you, Sarah. Good morning, bonjour. It's a great honor to introduce Professor Michael Meaney. He's James McGill Professor of Medicine at Douglas Mental Health University Institute of McGill University. He's also director of the Maternal Adversity, Vulnerability, and Neurodevelopment Project, and senior investigator at the Singapore Institute for Clinical Science. He is going to tell us about the latest development and perspectives in maternal modulation of neurodevelopment. Professor Meaney.
First, have a drink. Well, good morning, and bienvenue à Montréal. Okay, it's early in the morning, and I'm going to show you a fair number of rather detailed slides. Two things to keep in mind. First of all, there's a gist to the talk, and that's really what I'd like you to leave the room with. And second, of the 74 slides I'm going to show you, there's one that matters. So I'll take you to that. Just one. Okay, this one matters too. These are fantastic colleagues. I have a laboratory in Montreal and one in Singapore where I've worked for about 9 years, and these are colleagues in Singapore to whom I owe an enormous debt and who are remarkable contributors to the story you're about to hear. And this is essentially the take-home message, is that there is a profound influence of maternal mental health on fetal and postnatal neurodevelopment. It cuts across the population, and the one slide that I want you to take seriously is the one that suggests that this is not simply a problem of clinical dimensions, but is one of public health. So I'm going to walk you through primarily the results of a study, a longitudinal birth cohort study that we started 9 years ago in Singapore. It is a community-based study. We've recruited mothers from 2 large birthing hospitals in Singapore. There are 3 primary ethnic groups in Singapore. They are Han Chinese, Malays, and Indians. We have extensive antenatal phenotyping of the mothers, both in the area of metabolic health as well as in mental health. We have genotyped mothers and offsprings. We've been doing epigenotyping and extensive phenotyping of the offspring, including what will be the focus of today's talk, and that's magnetic resonance imaging. Now, I've shown this slide many times, and I suddenly started to tweak to the idea that there's a great number of people who are looking at this slide going, so where the hell is Singapore? What you're looking at, and I'm sorry, I'm pointer free at this, but if you look, Singapore is in red. It's right at the tip of the Malaysian Peninsula. It's a small island. It's sort of like Manhattan would be to New Jersey, kind of. And the bottom portion is Indonesia, and in Singapore we're squeezed in between. And so this study has focused on a number of dimensions, and I spoke to Sarah before giving this talk, and I said I could focus on the epigenetics or the neurodevelopment, and she said, well, they'd both be interesting, but the neurodevelopment would really, I think, be unique. So, of course, I start off with epigenetics. I want to give you this make one simple point. Most of the time, we're sitting around wondering how it is that individuals got to the point at which they are when we see them clinically. And we wonder and muse about the relative influence of genetic and environmental factors. And so that is, if you will, a primary concern we've been looking at, particularly in the context of fetal development. And we've used epigenetics in this particular case as a marker of phenotypic variation. The epigenome, as you've heard, I think, in your workshop, is perhaps the most rudimentary of phenotypic outcomes. And so we're studying this to start to understand how this particular phenotype may be influenced by relatively genetic and environmental factors. And so I'll walk you through just a little bit of this, but the bottom line is that even at this rudimentary level, there is a primary influence of gene by environment interactions. So the epigenetic mark we study is the most rudimentary of all. It is DNA methylation. As you know, over the course of early fetal embryonic development, epigenetic marks, particularly methylation, are laid down across the genome. Classically, DNA methylation associates with the silencing of gene expression. What happens is that the genome in certain regions acquires methyl marks. They attract a class of proteins known as methylated DNA binding proteins, which then in turn attract an entire complex of proteins that essentially suppresses gene expression. And the primary operator in this complex are histone deacetylases, which in fact bias towards closed chromatin. And this process silences gene expression by essentially working through the basic molecular mechanisms by which genes are transcribed. Transcription involves the binding of a class of proteins known as transcription factors to DNA binding sites, where they generally activate gene expression. When that region of the genome that normally binds transcription factors bears a methylation mark, that process is inhibited, and so the gene is silenced. So DNA methylation is classically associated with the silencing of gene expression, and one of the reasons why epigenetics has been so much fun to study is that it helps us to deal with what is a very fundamental problem in genetics and developmental biology, is how in the world do we create 220 different cell types from a single common DNA template, right? Every cell has more or less the same nuclear genetic material. We do so because these different cell types bear different methylation marks. And because the marks vary across the cell types, the patterns of gene expression vary, and hence you end up with stable phenotypic variation in cells that you find within the liver, the muscle, the brain, etc. Now, as it turns out then, methylation and other epigenetic processes can help us understand the process by which we create highly stable cell populations. And so if you were to look across the entire genome at epigenetic marks, and in particular at DNA methylation, you'd find that about 70% of the methylome does not vary across anybody in this room. Hopefully we all have livers, right? Pituitaries, muscle cells, etc. Well, those cells became those cells because of specific methylation marks, and they don't vary. But if you look in blue, you find a portion of the methylome, the epigenome, that can vary. And that portion, we think, is not so much involved in determining whether a cell becomes a liver cell or brain cell, but it might help us understand why the liver cell of one person differs from that of another individual. And so we think of that portion of the epigenome as being less stable and potentially vulnerable to environmental influences. And it is these type of processes that help us to understand what is in clinical practice a rather interesting phenomenon, and that is the discordancy we see in phenotypic variation between individuals who are identical monozygotic twins. And you know, it won't surprise you to understand that the mom of these two boys doesn't really have big trouble telling them apart, right? So what occurs despite the common nuclear genomes is a process by which epigenetically, perhaps through environmental influences, they differentiate. So what we've done in the context of the Singaporean study at one level is to simply ask what accounts for variation across these epigenetic marks in our cellular population. So we have a birth cohort. Through the heroic efforts of our obstetric collaborators, we were able to capture conceptual tissue in about 92% of the births. So we took umbilical cord and we surveyed the entire genome for DNA methylation. and we created maps of variable sites that differ between individuals. And we simply asked the question, what accounts for variation in these regions, those blue regions that do vary between individuals, these variable methylated regions? What accounts for that variation? Now, here's an interesting little statistical component. It gives you an idea of the degree to which, even at a rudimentary level, a phenotype as basic as DNA methylation can diverge from the underlying genome. This is a principal component analysis in which we took the genetic data, right? Not methylation, but genetic data from our 1,100 or so samples. And we ran, in this case, 200 or so. We ran a PCA analysis, and you can see the PCA analysis easily distinguishes the red Malays from the blue Chinese and the green Indian populations. So genetically, these are distinct populations. When you perform exactly the same analysis using DNA methylation, it's not quite so clear. You've created variation. You've gone from a population-based genetic to an epigenetic, and clearly variability has been created there. And our question is simply, what accounts for that variation? Well, in part, it's clearly due to genetic variation. Now, this slide is meant to tell you simply one thing. If you take all these variable regions that differ between individuals in methylation, and you map them against single nucleotide polymorphisms, what you'll see on this slide is that the overall distribution is skewed to the left, to very low p-values. That tells you just one thing. There is a relationship between underlying genetic variation and DNA methylation. So genes matter with respect to determining methylation patterns. But so too do environments. So here's what we have by way of a data set. We have 1,423 regions of the genome that vary between individuals in the level of DNA methylation. We have genotyping of the mothers and the infants, so we can look at the effects of individual polymorphisms. We have also phenotyped the mothers. We've looked at a wide range of potential environmental influences. We've got OGTTs. We can look at the mother's insulin sensitivity. We can look at birth outcomes, head circumference, birth weight, gestational age. We can look at maternal obesity, diet, and of course maternal mental health in the form of both symptoms of anxiety and depression. What you see in this pie chart is that in red is that portion of the 1,423 variably methylated regions that is best explained by the genetic SNP to which it's associated. Doesn't mean it's completely explained, but is best explained. The remaining 75% of these variably methylated regions are best explained by a gene-environment interaction between some feature of the maternal phenotype, be it maternal anxiety, depression, the birth weight of the infant, etc., and the genetic factor. If you actually account for ethnic variation, which tends to inflate the genetic component, that turns out to be about 15% genetic and 85% best explained by gene-environment interaction. And that, I think, is really the bottom line of this particular study, is the degree to which phenotypic variation, even at the level of DNA methylation, is primarily defined by an interaction between the genotype of the individual and their environmental circumstance. And that occurs early in life, including in the fetal period. The next slide is just to simply give you a proof of principle of this. And we did a study in which we looked at a specific genetic variation. This is a highly functional polymorphism in a gene that encodes for brain-derived neurotrophic factor. We looked at the interaction between that factor and maternal anxiety in defining the number of variably methylated regions. And what you see on the would be your left graph, in blue is a specific genotype and the effect of maternal anxiety. And you'll see the number of CPGs, variably methylated sites, that are associated with maternal anxiety varies as a function of the genotype of the infant. Hence, blue is substantially greater than green or whatever that other color is. And you'll also note is that that profile is unique to the infant's genotype. If you use the mother's genotype, you do not see that. So it's the infant's genotype that is moderating the impact of maternal anxiety on methylation. And that's a message that you'll see as we move into the MRI data. So essentially, bottom line, maternal mental health, amongst other features of maternal phenotype during pregnancy, matters enormously in defining phenotypic outcomes, and it does so by interacting with the genotype of the infant. Now, as far as maternal mental health is concerned, actually one of the individuals who graduated from our lab, Sonia Lupien, who's at Université de Montréal, has shown that if you associate maternal mental health over the postnatal period, over the first 7 years of life following birth, that you can see a statistical relationship between the level of depressive symptoms that a mother exhibits over that period following birth and the size of the amygdala, a brain region heavily implicated in mediating emotional processing and in our particular responses to aversive events. Mothers with higher levels of depressive symptoms have children with larger amygdalas, and you can see that from the graph in the middle, the scatter plot. On the right hand is a graph I'd like you to keep in mind, because we're going to come back to that particular theme, is that what Sonia was looking at, in addition to the size of the amygdala in these children, was their production of the stress hormone cortisol, which you'll note is also elevated amongst children whose mothers show a higher degree of depressive symptoms. And that will become relevant as we end this talk, because I want to talk about the potential influence of the stress hormone cortisol in defining variation in the amygdala, particularly over the course of fetal development. Well, one of the problems, there were a few proof of principle studies in which maternal mental health had been associated with neurodevelopmental outcomes in the offspring. One of the problems and challenges with that literature is that inevitably these studies were performed using children anywhere from 8 to 12, 14 years of age, which gave no indication as to what time in development this particular effect may be occurring. In parallel, there was emerging evidence which suggested that, in fact, it may well be the case that the primary influence of maternal mental health in neurodevelopment is actually occurring prior to birth, during the prenatal period. The question is how to test that, and the answer, of course, is that you image babies at birth. You do it right at the time or shortly after birth, in which you're able to document specifically the statistical relationship between mother's emotional well-being and child neurodevelopment. So what we've been doing in Singapore is fetal or neonatal neuroimaging. Kids are imaged, non-sedated, by the way. They breastfeed the kids, put them to sleep between 5 and 9 in the evening, and off they go into the scanner. We've imaged children shortly after birth, which is about a week or so, at 6 months of age, at 4.5 years of age, excuse me, now at 6 years and 7.5 years of age. So what do we see? Well, first of all, one of the challenges, as anyone who does neuroimaging will know, is that of actually defining an atlas for infant brain development. And so our colleague Chuan Ji at the National University of Singapore has met this challenge, defined an atlas, published. And so what we're able to do then is to analyze the imaging data in terms of volume of specific regions, microstructure using diffusion tensor imaging, as well as connectivity mapping. Now, here's the bottom line. When you look at the influence of antenatal maternal mood in variation in brain structures at birth, what you see is primarily an effect on a neural circuit, which is labeled here as a stress circuit, that involves, in particular, the right hippocampus, the right amygdala, and various structures in the prefrontal cortex. That is a network that is highly responsive to stressful events. It is also the network that is most strongly implicated in clinical disorders of depression and multiple forms of anxiety disorders. And interestingly, it is very specific largely to the right hemisphere, particularly with respect to the amygdala. What we see here is that I will show you evidence for the idea that both at the level of volume, particularly at the level of microstructure, and even at the level of connectivity, there is a profound influence of maternal mental health on this particular network. Now, one of the things that I must admit was really quite striking is the specificity of this particular effect over the course of prenatal development. What you're looking at here is a summary of a wonderful meta-analysis performed a number of years ago by Mir Etkin and Richard Wagner, looking at functional imaging studies in patients with various forms of anxiety disorders. Now, this is patient data, so these are clearly adults. And what they surmised was that these functional imaging studies most commonly focused on and revealed a network that went from the right amygdala to the insula cortex, and then started to recruit other regions within the prefrontal cortex. But specifically, the regions that emerge in adult studies of anxiety disorders are the right amygdala and the insula. What we see when we look in neonatal imaging studies is a selective effect of maternal depressive symptoms, or anxiety symptoms, by the way, on the right amygdala. And in this particular case, we're looking at diffusion tensor imaging, so the microstructure of the right amygdala differs significantly between mothers with high versus low levels of depressive symptoms. When we do connectivity mapping from the right amygdala, what you find is significant differences in connectivity revealed by functional imaging between the right amygdala, specifically the right, not the left, and the insula. In other words, that same network that reveals itself in anxiety studies of adult patients using fMRI is apparent and reflecting the influence of maternal emotional well-being already at the time of birth. And what happens, in fact, is that you're actually advancing the connectivity. So there is greater evidence of connectivity between the right amygdala and the insula amongst infants born to mothers with higher levels of either anxiety or depressive disorders. So it's an example of accelerated development, and colleagues, particularly Nim Tottenham now at Columbia and Regina Sullivan at NYU, have used both rodent and human models to show that in postnatal development, adversity accelerates the maturation of these corticolimbic connections. So it is, if you will, consistent with the idea that exposing a child to early life adversity associates with early onset of maturity of these particular circuits. There is, in fact, evidence for a persistence of these effects. So when we image the children at 4.5 years of age, and we look at either evidence for prenatal or postnatal depressive symptoms, we can see statistical relationships for both with the size of the right amygdala. And so what you're looking at here on the left is the association in boys and girls at the time of birth between depressive symptoms and the size of the right amygdala. Controlling for postnatal, well, this is at the time of birth, but if you do the postnatal study at 4.5 years of age, looking at concurrent depressive symptoms, in the middle graph, you see the association across both boys and girls. On the left-hand side, right-hand side, what you see is the selective association between postnatal depressive symptoms in the mothers and amygdala development in girls. This effect actually persists, and it does so far more prominently in girls than it does in boys. While the gender specificity is somewhat mystifying, it is entirely consistent with what we know about the transmission of the risk for maternal depression to that of the offspring. It is a statistical relationship that is far stronger in daughters than it is in sons. So the gender specificity of this is intriguing, but what's really most important, of course, is the idea that these effects are persisting and reflecting both prenatal and postnatal effects. Now, the next question then, of course, is so what? The brain imaging is wonderful, but does this really map on to what we see later on when we look at socio-emotional development in children? What you're looking at here in the top panels is the association between the right amygdala microstructure and socio-emotional problems as identified by the Child Behavior Checklist at 2 ages, 24 on the left and 48 months on the right. What you see at 24 months is that the right amygdala is significantly predictive. Now, the thing to keep in mind, this is amygdala, and in the bottom panels, insular dimensions at birth. So the right amygdala microstructure at birth predicts socio-emotional problems at 24 months of age. Less so at 48 months of age. What you see when you look at the insula in the bottom panels is that there is no relationship between the insula structure at 24 months, but by 48 months of age, there has emerged a very profound influence. So in a sense, what you're seeing is evidence of cerebralization, where these prefrontal regions are actually starting to become predominant. Nevertheless, what you also see is evidence that even using brain imaging at birth, it is predictive of these socio-emotional problems later on in life. Now, when you're collecting mounds of data, such as with MRI and behavioral outcomes, it's often fun to hand your data over to a mathematician whose only knowledge of the brain is basically that it's somewhere north of the pituitary. What this allows you to do is to use completely unbiased approaches to defining the relationships that you think exist a priori. So we handed this over to Wee, a mathematician who used a cluster, an unsupervised cluster analysis. What he's doing is to take all our neuroimaging data across individuals, 200 or so individuals, and to create cluster maps. These are simply maps where specific measures correlate highly and thus cluster, without any knowledge of what those underlying parameters may be. So he creates a cluster map, and in doing so, unearths 4 individual clusters. Now, the cluster at the top, as you can see, is a rather weak one, but clusters 2 and 4 in particular, as well as number 3, are statistically quite strong. So we then turn around and say, okay, we don't know anything about what underlies or defines these clusters, but let's see if they are predictive of childhood behavioral problems at 24 and 48 months. And what you see here are the CBCL scores for various scales. What you'll note in particular, if you look on the right-hand side, is that cluster 4, children defined by cluster 4, show greater evidence of socio-emotional problems at 24 months than do kids defined by cluster 2. And that same relationship persists at 48 months. Again, keep in mind these children have been clustered on the basis of brain imaging data that was acquired shortly after birth. Then what you can start to do is to decompose these clusters and to ask what element in the cluster is most predictive of those particular features. And the answer is, it turns out to be the amygdala and largely the insula. So even coming at this data set from a completely unsupervised way aligns the connectivity between the amygdala and the insula with these socio-emotional problems. And again, to underscore the major point here, is that this particular relationship between the amygdala and the insula is one that later on comes to define individuals with anxiety disorders. Here's the slide that counts. What I've spoken to you about is data from a community sample. Virtually all of these studies are drawn from a population that varies in depressive or anxious symptoms. What you're looking at here is the distribution of all the mothers in our Augusta cohort studies as defined by the Edinburgh Postpartum Depression Scale. 15 is generally the cutoff that is used to define mothers who are pregnant who are probable for depression. That's what you screen for in obstetrics. 9.5% of our Gusto population screened thus positively. A little bit lower than what we might find in the United States or Canada, but very much in the same range. However, 29% score in the subsyndromal range. That is high levels of depressive symptomatology, but not quite at the level of clinical dimensions. That is defined in our study by scores of 9 to 15. In other studies using these same metrics, what you find is that the mothers who score in the subsyndromal range versus and compared statistically to those in the clinical range do not differ on measures of psychosocial dysfunction, parenting of their children, or most importantly, childhood outcomes. The point here is, if we're talking about maternal mental health as a problem with respect to child neurodevelopment, it is a problem that can be reduced to at least 40% of the population. It is not unique only to those mothers with probable clinical symptoms. It in fact encompasses almost half of the mothers that we see. And that is a problem that I think brings this into the scope of enormous public health concern. Now, I've spoken primarily about the influence of maternal emotional well-being over fetal development, simply to alert you to the idea that there is strong evidence that these same factors operate postnatally, and so there continues to be influence of maternal factors operating obviously after birth. What you're looking at here is just simply evidence that one can also associate postnatal symptoms of maternal anxiety, independent of prenatal symptoms, in terms of looking at these same neurodevelopmental outcomes, particularly looking at both the hippocampus and the amygdala. And Joan Luby has recently published a wonderful longitudinal paper of imaging data in PNAS showing that maternal nurturing forms of maternal care, such as social support, enhance the postnatal growth, in this case, of the left hippocampus. So these influences clearly persist beyond this point in time, and one can then immediately jump to what might be the plausible clinical implication that what one sees over the course of prenatal development may be subject to postnatal reversibility. Okay, I want to bring you back to the theme I opened with, which is that of gene by environment interaction. I mentioned when we study the epigenome, for example, we see clear evidence that phenotypic variation is in fact largely a product of an interaction between the gene and environment, and in fact that's no less true when we look at imaging studies. If, for example, we take neonatal imaging, we focus on those structures that vary meaningfully in relation to many of our environmental constructs, notably either microstructure using diffusion tensor imaging in the amygdala or hippocampus, or volumetric analysis in these same regions. When you do this same type of gross analysis, aligning these metrics to either polymorphism, SNPs, single nucleotide polymorphisms that reveal genetic influences, or gene by environment interactions, there's actually an interesting consistency here, whereby measures of volume seem to greatly reflect genetic variation and some contribution of gene by environment, but the more dynamic measure of microstructure, which we would think of as axonal outgrowth and synaptogenesis, is largely emerging as a product of gene by environment interaction. And we've drilled down on this in terms of looking at a specific proof of principle. And in this particular case, we looked at haplotypes, genetic variations that define the function of a specific gene, catechol-O-methyltransferase, which is a gene that produces a degradative enzyme for dopamine, largely in the prefrontal cortex. So what we're looking at then is genetic variation in the gene for COMT and its interaction with maternal anxiety. And what you find essentially is a rather interesting pattern. I'll just skip to what is actually the bottom line here, is that if you look at infants bearing functional polymorphisms in the COMT gene, is that that genetic polymorphism defines both the magnitude of the impact of maternal anxiety, and it also defines which regions within the cortex are affected. And so, for example, if you look at a specific haplotype, you can see that there is, first of all, a lesser or greater effect of maternal anxiety, but you can also see that the brain structures that are affected by maternal anxiety also vary as a function of the genotype of the infant. So it is clear that regardless of the effect of maternal emotional well-being, that the underlying genetic variation is defining both susceptibility to maternal emotional well-being, as well potentially as which structures are actually affected. Whoops. Okay. Now, I want to then take you vastly into the area of speculation because one of the reasons why genomics is really interesting to use as a tool is that it has the potential to actually reveal what might be candidate biological mechanisms that underlie the relationship between maternal emotional well-being on one hand and child neurodevelopment on the other. And I want to give you an idea of how we do this. So we have actually performed GWAS, genome-wide association studies, associating the genotypic variation of the infant to patterns of neurodevelopment. Our goal, by the way, is certainly not to try to uncover critical variants that might be associated with such particular outcomes. There's no question that when you look at anything as complex as the development of the amygdala, the hippocampus, or certainly prefrontal structures, is that that involves many thousands of genes operating within networks to basically underlie variations in multiple biological systems. So the question is, can we use genomics to help us identify those underlying systems? So let me tell you the way we've done that. We've done that by using what are referred to as polygenic risk scores. So within the area of genomics, we've moved off the idea that we're going to find smoking guns for common disorders. In the area of depression, for example, the assumption is that the genetic burden that biases individuals to depression is going to be defined by variants in probably many thousands of individual genes. So the question then becomes, can we create a cumulative index of those relevant genetic variants? So we've gone into using large GWAS studies to define those genetic variants that show a nominal statistical relationship to depression. We cluster them together and we say, these are the guys most likely to define your risk for depressive symptoms. And we call that a polygenic risk score for major depressive disorders. What we then did was the following. We said, well, look, one of the reasons why a cumulative genetic risk for depression may bias you towards that particular disorder is that it interacts with relevant environments to define the neural circuits that are ultimately involved in depression. And we tested that idea. So we took our neonatal imaging data and we looked at two environmental conditions that we know influence the development of areas like the amygdala and the hippocampus, as well as prefrontal cortex, over the course of prenatal development. And that was, first of all, of course, antenatal maternal depressive symptoms. It was also socioeconomic status. And what you find, if you look in both of these particular graphs, is that if you look at the right amygdala again, you see that the association between maternal depressive symptoms or socioeconomic status and right amygdala volume is moderated by the genetic score for depression. So infants with a higher polygenic risk score for major depressive disorder are more susceptible to either the conditions of poverty or to higher levels of maternal depressive symptoms. Now that cumulative risk score is made up of many thousands of variants, okay? And what you've said is that score that reflects these many thousands of variants moderates the impact of these two relevant environmental conditions. you can then hunt within those many thousands of variants and say which ones best moderate that association between either maternal depressive symptoms and right amygdala or socioeconomic status in right amygdala. So you're just statistically trying to unearth which variants have the biggest moderating effect. When you take those particular variants that you've identified, you can then map them onto genes, right? And you can then ask, do those genes form specific biological processes? And the case in this particular instance is they do, in particularly in the case of the right amygdala. In the right amygdala, when you unearth those genes that were involved in your polygenic risk score for depression that best moderate the association between either maternal mental health or socioeconomic status, they cluster and form a specific biological process, which is known as the metabotropic glutamate receptor pathway. Now, the interesting thing about this was that we had actually replicated this finding in a US sample based at Irvine in collaboration with Claudia Buss. So in both cases, we were able to demonstrate the association between maternal depressive symptoms and SES and right amygdala volumes. So we've got two cohorts in which to look for those particular outcomes. And in fact, what we find is that when we look for this particular metabolic glutamate receptor pathway, we see it in both the Irvine sample as well as in the Singapore sample as the most highly statistically probable biological pathway. Now, when you look at the network that you've identified using these genes, it in fact is a network that involves glutamate and the release of glutamate, which should surprise no one. It involves the snare complex, which is involved in creating synapses, and in vesicular processes, which are also involved in that. So essentially what you've said is that it appears probable that maternal depression is regulating amygdala development through its ability to alter glutamatergic signaling in the amygdala and downstream processes of amygdaloid synaptic development. Okay, I'm going to leave you with one last point. I want to take you very briefly back to work on epigenetics. And in doing so, I want to give you a hypothesis for how the effect of maternal depression and glutamate-mediated synaptic plasticity in the amygdala might be occurring. So we've talked about the importance of the amygdala as well as the hippocampus in this stress circuit. We've also looked at socioeconomic status, and what we've defined in doing socioeconomic status is an area of the genome in which DNA methylation is statistically associated with socioeconomic status. So this goes back to the umbilical cord samples of which I spoke to you way at the beginning. We see that there's a statistical relationship between socioeconomic status of the mother and variably methylated regions in the offspring. We can then take those methylated regions and we can then map them and ask, is there something distinct about those particular regions? Now, when we do that, we know, of course, that socioeconomic status is a very broad macro variable. If you look in terms of developmental outcomes, and particularly those that involve brain-mediated phenotypes, there are largely two processes that are associated to socioeconomic status that mediate the influence on neurodevelopment. They are, first of all, birth outcomes, such as fetal growth and development, and second, maternal mental health. We can then pull out those variably methylated regions that statistically associate with socioeconomic status and are also linked to maternal mental health. And when we do so, when we take those methylated regions and map them onto genes, what we find is that they inform us about a specific biological process. And this is pro-opiomelanocortin pathway, or POMC pathway. The reason the POMC pathway is relevant is that it is a central feature of the activation of adrenal glucocorticoid release. So cortisol is released from the adrenal gland in response to adrenocorticotrophin, which is made from pro-opiomelanocortin. And if you mentioned earlier when I talked about Sonia Lupien's data, I talked about the potential relationship between cortisol and the amygdala. So this network associates with enhanced activity of the pro-opiomelanocortin. When we do GWAS studies, looking again at the amygdala, asking what statistical variants, what genetic variants associate with individual differences in the amygdala, we unearth a network that is anchored by a gene known as NR3C1, also known as the glucocorticoid receptor. So what you have are two converging lines of evidence. One through methylation, which suggests adrenal glucocorticoid mediation, and another through genetic variants that suggests that the glucocorticoid receptor gene is a hub for genetic variants that associate with amygdala volume. And in fact, Claudia Buss, who's now at La Charité in Berlin, had provided direct evidence of this in an earlier study in which she used amniocentesis to define maternal cortisol levels and showed that mothers with higher antenatal levels of cortisol in their amniocentetic samples had larger amygdalas. And so essentially, what you have is information derived from these particular sources suggesting that glucocorticoids are mediating this particular effect. I mentioned to you earlier that the glutamatergic pathway seemed to be one that was heavily involved, and glutamate release is tightly regulated by glucocorticoids. Now, all of that, of course, is a matter of speculation. It is simply an illustration of how these data sets can actually inform us with respect to these pathways. What I think has moved beyond the matter of speculation is, in fact, the profound influence of maternal mental health and the degree to which we need to consider this as a profound public health issue. Thank you very much.
So we're going to have questions after the two speakers. Our next speaker is Dr. Robin Ohls. She is a professor of pediatrics and chief of neonatology at the University of New Mexico. She graduated from Stanford University and completed medical training at the University of Utah. And that has provided an ongoing research collaboration between the neonatologist where I work and myself in the neonatal follow-up clinic and Dr. Ohls. She's performed clinical translational research for 25 years, has focused on the benefits of erythropoiesis stimulating agents in neonates. Dr. Robin Ohls, thank you.
Thank you so much, Sarah. Thank you for inviting me to this conference to share our research, our combined research together. Make sure I get these right here. I have no conflicts of interest to disclose, and I will be discussing non-approved use of erythropoietin, or EPO, and Darbepoetin, or Darby. I'd like to review with you today some of our translational research showing the effects of erythropoiesis stimulating agents, or ESAs, on fetal and neonatal progenitors, and discuss recent studies evaluating potential neuroprotection of ESAs in preterm infants. So as a neuro-focused group, you may know more about ESAs and neuroprotection, but many in the neonatology community don't really understand this connection yet, and so we are trying to investigate it and provide that information. Now, erythropoietin is a 34 kilodalton glycoprotein with 165 amino acid backbone chain. It was cloned in 1985, and very soon after that, in 1989, was approved by the FDA to treat adults with end-stage renal disease and anemia. Amgen Pharmaceuticals, which manufactured EPO, investigated the relationship between the sialic acid containing carbohydrate content and the serum half-life of this protein. And they were able to biologically modify EPO by changing 5 of the amino acids to allow for increased carbohydrate binding sites. This provided a longer serum half-life and a greater biologic activity, and the FDA approved Darbepoetin, or Darby, in 2001. We've been evaluating EPO in preterm infants and also in term infants since the early 1990s, and we wanted to know if Darbepoetin in vitro also affected red cell progenitors in the same way that EPO did. We were able to isolate fetal progenitors from marrow, liver, and from both term and preterm cord blood. We cultured these cells with various concentrations of EPO or Darby and looked at erythroid colonies at 12 to 14 days. We also quantified the number of normal blasts or individual cells per colony. Here are some images of different sizes of colonies. On the left, you can see multiple smaller normoblast colonies. In the middle, some medium-sized colonies. And on the right, some large erythroblast colonies. I'm showing you the results of the bone marrow progenitor response to EPO and Darby. The liver progenitor response and cord blood progenitor response show similar graphs. What we identified was that although it required a slightly increase in protein equivalent concentrations of Darby, erythroid progenitors were sensitive to both erythropoietin and Darbepoetin in a dose-response fashion. When we looked at cord blood progenitors, in fact, it appeared that preterm cord blood progenitors were even more sensitive than term cord blood progenitors to Darbepoetin. This gave us the evidence that suggested that if we used Darbepoetin in preterm infants, we could stimulate red blood cell production. Over the years, other investigators have looked at other effects of ESAs that include neuroprotective effects. And while we've known about the erythropoietic effects of EPO and Darby for quite some time, it's only been in the last 10 to 15 years that the neuroprotective effects of EPO and Darby have been identified, mainly in animal studies. And in fact, both EPO and Darby work primarily by decreasing apoptosis, both in red cell progenitors and also in neuronal progenitors. So it stimulates neurogenesis, oligodendrogenesis, it prevents apoptosis in the brain, and also prevents injury due to excitotoxic and oxidative injury. We've looked at these effects in neuronal cells during fetal development. This is human fetal development. We were able to take neuronal cells and culture them, again, in different concentrations of both EPO and Darby. And in fact, did see a dose-response relationship between the number of neuronal cells grown in culture with increasing concentrations of both erythropoietin and Darbepoetin. And it appeared, in fact, that perhaps Darbepoetin was slightly more neurogenic than EPO at similar concentrations. When we looked at the individual cells in these cultures, we identified a number of different phenotypes, primarily astrocytic in nature, but also a number of neuronal progenitors and some mixed progenitor cells. And when we looked at gene expression of these cells, we identified a significant difference in the effects of both EPO and Darby compared to control cell populations, in that nestin gene expression, which reflects neuronal progenitors, was significantly increased in those cells that were exposed to Darbepoetin compared to EPO and compared to control cells. And it appeared that Darby may have more neurogenic effects than EPO. Of course, this was in the laboratory, and we needed to prove this by giving this to babies. So I wanted to walk you through the clinical studies that we performed since really the early 1990s. But we began this study in 2005 in collaboration with the University of Colorado and the University of Utah. We randomized 99 preterm infants that weighed 500 to 1,250 grams to one of 3 treatment arms, either Darbepoetin, given at a dose of 10 micrograms per kilogram once a week, EPO given at 400 units per kilogram 3 times a week, and this is considered a hematopoietic dose of EPO, or a placebo arm that received sham dosing during their hospital stay. Infants were treated for 35 weeks, through 35 weeks corrected age, and we followed them in the hospital and then developmentally at 18 to 22 months. The primary outcome that we focused on during the hospital phase were evaluating the number of transfusions that these infants received. We also powered the study to evaluate a primary outcome at follow-up of a difference in cognitive scores between these groups. During the hospital phase, compared to the placebo infants, those infants that received either EPO or Darby, which we call those the ESA groups, received fewer transfusions, about half the number of transfusions than those in the placebo group, and were exposed to fewer donors. And this was the first study to show actually a decrease in donor exposure in premature infants given ESAs. Infants receiving ESAs also had a higher hematocrit and higher reticulocyte counts, which we use as evidence of active erythropoiesis. And there were no differences in these groups in the common morbidities that we see in preterm infants, including retinopathy of prematurity. When we evaluated these infants at 2 years of age, we saw that there was a significant increase in cognitive scores in those infants in the Darby and EPO groups compared to the placebo group. In fact, there was also an increase in executive function scores, and the infants in the Darby group had higher executive function scores than those in the EPO group. Both the ESA groups had higher scores than those in the placebo group. And all of this added up to improved neurodevelopmental outcomes, so there was an increase in neurodevelopmental impairment in the placebo group. This was primarily driven by the fact that of the infants that had cerebral palsy at 2 years of age, all those infants were in the placebo group. None of the infants in the ESA groups had any evidence of cerebral palsy. We were able to continue following these infants in an NIH-sponsored study termed the BRITE Study, or Brain Imaging and Developmental Follow-up of Infants Treated with Erythropoietin. We hypothesized for this study that infants that had received ESAs during their hospitalization would have improved long-term neurodevelopmental outcome and regional brain structure. All those infants that were enrolled in the original study were eligible. In addition, we enrolled a term cohort control. Those are infants that were born at term around the same time of year and age, but had been able to go home from the hospital without any ill effects. We provided developmental testing and MRI imaging at 3.5 to 4 years of age, and again at 5.5 to 6 years of age. In this group of a subset of the original study, again, we saw an improvement in the full-scale IQ score in the ESA group compared to the placebo group. The term infants scored better than both the ESA group and the placebo group. In terms of executive function measured in these infants, those that were in the ESA group had improved into executive function, as we identified at 18 to 22 months, and improved working memory. Again, the infants that had been born at term scored better than the preterm infants in all of these categories. When we follow these children now out to 6 years of age, so now these are early school age children, again, there was a significant difference in full-scale IQ and performance IQ between the ESA groups and the placebo groups. There was also improvement in cognitive flexibility, one of the executive function tests performed at 6 years of age between the ESA groups and the placebo groups. Importantly, note at this point in time, the difference between the ESA group and these term controls had now disappeared. So we were able to have these premature infants who otherwise would score behind term infants throughout preschool and school age now catch up to their term cohort controls in terms of all of these cognitive outcomes. There have been a number of other clinical studies looking at neuroprotective effects of EPO in preterm infants. I'm not going to review the term infant studies today, but there are also term infant studies looking at the effects of EPO and Darby on infants with HIE that are being cooled. There was a large randomized controlled trial in Switzerland performed approximately 5 years ago now, where infants less than 32 weeks of age without grade 3/4 IVH were randomized to receive high-dose EPO for a total of 3 doses in the first 2 days of life. A total of 443 infants were randomized in this study. Infants' gestational age ranged from 26 to 32 weeks. The hospital phase of this study reported that of those infants enrolled in the group, there were no difference in the mortality between the groups and no difference in the common morbidities seen in premature infants. It appeared that this dose of EPO, which was 3,000 units per kilogram, so approximately 10 times the dose that we generally use, was safe given in this form. There were a few studies looking at MR outcomes in these infants at term. 3 of the sites performed MR imaging and found that there was a decrease in white matter injury, white matter signal intensity, and periventricular white matter loss and gray matter injury in those infants in the placebo group compared to the EPO group. One of the sites also was able to perform DTI in these infants and showed that there was improved mean FA in the EPO group compared to the placebo group, and they concluded that EPO improved white matter development in preterm infants. The developmental outcomes of this study have not been as satisfying as our previous study. When they looked at these infants at 2 years of age and performed the Bailey Scales of Infant Development 2 scores, they showed that there was actually no statistical difference between these 2 groups of infants. One of the issues with this study was that the infants in this study were larger than the infants that we had previously studied. They excluded infants that had any sort of significant interventricular hemorrhage at the time of birth. And so these were a relatively healthy population of preterm infants that were being compared with the EPO and placebo groups. A similar study was performed in China, randomizing, again, infants less than 32 weeks gestation, excluding infants with grade 3/4 IVH. In this study, 800 infants were randomized. They received a dose of EPO of 500 units per kilogram every other day for a total of 2 weeks, or placebo. The primary outcome of this study was death or moderate to severe neurological disability at 18 months corrected age. There was a significant difference between the EPO and placebo groups in this study, in that over twice the number of placebo infants either died or had significant neurologic impairment at the time of evaluation. This was largely driven by the significant amount of grade 3/4 IVH that was identified in these infants. And again, at the beginning of the study, infants with grade 3/4 IVH were excluded. So these are additional infants that experienced a grade 3/4 IVH during the study. Interestingly, the number of infants that were transfused were lower in the EPO group than the placebo group in this study. That was not seen in the previous Swiss study. And again, the incidence of ROP was not different between these 2 groups. These investigators concluded that repeated low-dose EPO in these infants reduced the risk of long-term neurological disability without any obvious adverse effects. So why aren't we using ESAs in all of our preterm babies? Well, the data are still being collected. Evidence is still being built in order to support the use of ESAs in preterm infants. And we believe that the neurodevelopmental improvements with ESAs is a multifactorial process. It's not just the biological effects of ESAs that have been shown in both animal models and in vitro in human developing tissue, or decreased transfusions, but it's a combination of all three of these. We know that, that there is an improved neurodevelopmental outcome as evidenced by improved cognition in our infants, which is associated with higher peak and trough EPO concentrations. So when we measure EPO peak and trough concentrations and Darby peak and trough concentrations in our preterm infants, we identify a sort of a cutoff level of about 500 milliunits per mil of EPO in the serum. This is an elevated concentration, so anybody here probably would not have that level of EPO in their blood unless they had actively hemorrhaged right before they came. But in our infants, this level was associated with neuroprotection and with improved cognitive scores. The relationship between cognitive scores and EPO peak and trough concentrations is shown on this slide. So on the left is full-scale IQ in relation to EPO peak concentrations. On the right, full-scale IQ with trough concentrations. And there's a significant relationship with both peak and trough concentrations. Because of the prolonged half-life of Darby in these infants, we believe that this may, in fact, be some of the benefit that we can see with Darby over EPO in our preterm infants. We also have evidence that improved cognition is associated with decreased white and gray matter injury and improved FA, especially in females. This is data published by Dr. Phillips, who is actually the brains of the operation, I would say, in our group, showing that the FA is improved in the ESA group over placebo, but this is primarily driven by improvement in females compared to males. And the associated decreased white and gray matter injury that was identified in females was focused on these four areas of tracts: the cingulum, anterior corona radiata, the superior longitudinal fasciculus, and the inferior frontal occipital fasciculus. In addition to the biology that we have studied, looking at mechanisms of neurodevelopmental improvements, we know that having an increased red cell mass results in an increase in hemoglobin concentration in our infants, which may increase oxygen that is available to tissues, especially the brain. And when we look at the number of EPO studies that have occurred over the years, this is data from our study, but in all studies, despite the fact that we try to maintain hemoglobin concentrations similar in both placebo and EPO groups, in fact, those infants that received EPO and Darby in our group, reflected by the red and blue lines here on this graph, had significantly higher hematocrits throughout the study. So we used transfusions as a way to keep the hematocrit up in those infants in the placebo group. Those infants in the Darby and EPO groups received fewer transfusions, but were able to maintain their hematocrits at a higher level. Finally, we believe that those infants that received fewer transfusions actually have improved neurodevelopmental outcomes, regardless of receiving EPO or Darby. When we look at cognitive and language scores in our infants at 2 years of age and compare the number of transfusions those infants received, infants that received no transfusions, whether they were in the placebo or ESA groups, had similar cognitive scores and language scores. But any infant that received any transfusions in the placebo group had a significantly lower cognitive score than their counterparts in the ESA group. In other words, ESAs may have provided some sort of protection against some of the adverse effects of transfusions that we see in preterm infants. So to summarize the data from our ESA studies, similar to EPO, we identified Darby effects in the lab that both preterm and term erythroid progenitors, also progenitors from liver and marrow, were sensitive to Darby as well as EPO. And in fact, Darby may increase the number of neuronal progenitors that we can find in babies. Preterm infants respond to ESAs with an increase in hematocrit, a decrease in the number of transfusions, and decreased donor exposure. We know that peak concentrations are higher after Darby dosing compared to EPO, and that ESA concentrations correlate with cognitive scores at both 2 and 4 years of age. Finally, ESAs administered to preterm infants result in improved neurocognitive outcomes at 2, 4, and at 6 years of age. Based on our previous studies, we've been funded by the NIH to perform, hopefully, my last randomized placebo-controlled trial of ESAs in preterm infants. Our primary hypothesis is that preterm infants administered Darbepoetin will have improved neurocognitive outcomes at 22 to 26 months corrected age compared to placebo recipients. We plan to enroll 325 infants per group for a total of 650 infants to determine a difference of 7.5 points in a standardized score of the Bayley Scales of Infant Development 3 cognitive test with 90% power. In our studies, we enroll multiples, so twins and triplets, into the same study arm. We feel that it's difficult for parents to make that decision to consent to a study and then randomize their infants to different arms of the study. And we anticipate a 75% survival in this group of infants. We'll be studying infants that are on the very limits of viability in the NICU, so infants that are 23 and 0/7 to 28 and 6/7 weeks gestation. These are our smallest and most vulnerable premature infants. They'll be randomized in the first day of life to receive Darby. Again, we'll use the dose of 10 micrograms per kilogram, given either subcutaneously or intravenously, or placebo. In our babies, we can give IV placebo, but we don't give subcutaneous placebo shots. We use sham dosing. All of the infants receive either IV iron or oral iron supplementation, and this is an important part of maintaining improved neurodevelopment in our preterm infants, most of whom are iron deficient. Infants will be transfused in our study using specific guidelines, and we'll evaluate their neurodevelopment at 22 to 26 months corrected age. We'll look at a number of secondary outcomes in our study. Again, we follow transfusions and donor exposures during their hospital phase, look at the hematologic indices, and we'll actually measure the red cell mass in our babies. We always follow the comorbidities. Our preterm infants are a high-risk population, and they have a baseline morbidity rate that's significantly higher than term infants. And we look at a variety of morbidities, including retinopathy of prematurity, intracranial hemorrhage, sepsis, bronchopulmonary dysplasia, and patent ductus arteriosus, and necrotizing enterocolitis. We'll look at Darby peak and trough concentrations and evaluate pharmacokinetics in our population. We also plan to look at biomarkers for brain injury and recovery, including GFAP, S100B, and activin A, and look for markers of inflammation, including TNF-alpha and a number of interleukins. This is a study that's actually going to be started this month through the NICHD Neonatal Research Network, and we anticipate that we will have results probably in about 5 years. So I'd like to finish by thanking my BRITE study team. Dr. Phillips is here in the audience today, and thank our funding sources, which were initiated through the Thrasher Research Fund, and then followed through with NICHD and NHLBI. Thank you very much for your attention.
Thank you so much to both of our speakers. I'm hoping that you're all formulating some questions for them. There was a lot of material within that would certainly generate a few questions. Thank you, Dr. Chambers. Go ahead.
Sorry. Hi, I'm Hank Chambers. It's on. I'm Hank Chambers. I'm a pediatric orthopedic surgeon, so this was stretching my mind. Dr. Meaney, I wanted to ask you about the kids, the mothers who have depression. Have you ever done any interventions? So if either a hypothesis or children, mothers who have actually been treated with antidepressants or therapy or anti-anxiety agents, does it change the epigenetics of the children?
That's obvious. Am I on? Yeah, too much. That's obviously the key question. So I'm just walk you through because it's obviously the important question. We have not done it. There are studies out there, particularly from Myrna Weissman's laboratory in Columbia, in which they've used psychotherapeutic interventions with the mothers and show that it offsets the later risk, particularly for depression in the offspring, but also if you move somewhat more earlier in development for socio-emotional problems. So there is, I think, good evidence that psychotherapeutic approaches, non-pharmacological approaches, offset the risk for the offspring with respect to depression or depression-like phenotypes. The reason I'm being specific to non-pharmacological is that there is emerging evidence that the SSRIs, the selective serotonin reuptake inhibitors that are commonly used, all too commonly used, to treat either anxiety or depressive symptoms, are themselves associated with some toxic effects with respect to development. So they are not, I don't think, the good answer to the question of if we do something about the mother's mood, will it improve outcomes for the child? I think that lies more within the realm of psychotherapy. And then I'll just take you into the realm of speculation. The question, if you turn to people in the public health sphere, is that if you're talking potentially about as many as 40% of the mothers who would benefit from a particular intervention, how in the world are we ever going to deliver that? And there is emerging evidence, it's preliminary, but it's encouraging, that therapy-assisted, web-based psychotherapeutic interventions using things, traditional cognitive therapies, interpersonal behavioral activation, actually has a beneficial effect with respect to the mother's mental health. So I've given you a broad scope here. The answer, simple answer to your question is yes, if you're not talking about SSRIs, which have their own complications, improving the mother's emotional outcomes improves the neurodevelopmental outcomes for the child. And the emerging evidence, I think, suggests that we might be considering how to use web-based technologies to try to reach that segment of the population.
But that hasn't really been studied yet. That's just your
No, it
hypothesis.
has not. And it had the web-based, the psychotherapeutic interventions have been studied extensively with respect, but that's face-to-face psychotherapeutic interventions. The web-based has only preliminary evidence of impact on the mother's emotional well-being.
Great, thank you.
Pleasure.
Mike, go ahead.
Michael Massal, University of Chicago. Erythropoietin was discovered at University of Chicago, but I have no conflicts of interest. This is a question for Dr. Ohls. One of the things that you make me think of is the fundamental wisdom of connecting basic and clinical and translation sciences to a biomarker like hematocrit and erythropoiesis. And one of the things that I wonder about, because of the complexity of transfusions and things, are some of these infants having placental transfusions at birth, what's known as cord milking or cord extra time? And would you comment on that?
Well, thank you for that question. I would say that a majority of NICUs, at least in the United States, are using delayed cord clamping or cord milking as part of the initial resuscitation process for those infants that are not critically ill or significantly depressed at birth. So infants are starting out, thankfully, with slightly higher hematocrits than 10 or 15 years ago when we initially started our studies. That being said, the volume that we're dealing with in the babies this size and this gestational age still is somewhere between 45 cc's and 80 cc's total of blood. And so it's literally trying to monitor every single drop of blood that is drawn from the baby and trying to improve red cell mass by stimulating erythropoiesis. But I would say most of the babies are getting delayed cord clamping now, yes.
And the second comment I would have is this study that you currently are doing for infants with 23 to 26 weeks gestation is tremendous in its potential, because historically studies of intervention have only focused on the biological non-CNS endpoint so as not to make disabilities worse and not to study neuroprotection. You are to be praised for including that as an outcome measure, empowering your study to do that. Thank you.
Thank you.
Thank you, Mike. Barry?
Dr. Meaney, Barry Russman, Portland, Oregon. At what time, and maybe I missed this, but at what time in the pregnancy were the mothers identified as being depressed, and how were they identified as such?
Yes, no, you didn't miss it. I'm afraid I failed to mention that. In the initial study, we used a singular assessment of depression at 24 to 26 weeks, so second trimester, late second trimester. It was done using a battery. So we used the Beck Depression Inventory. We used the Edinburgh Postnatal Depression Scale, which is validated for antenatal use, as well as the State Trait Anxiety Inventory. And we've actually done a fair amount of psychometrics looking at sub-properties of those individuals. What we have done subsequent to that is a follow-up cohort study in which we've actually tracked maternal symptoms, and this was actually a preconception study in which we recruited women who had expressed the intention to become pregnant. We used the same tools to assess maternal mental health prior to conception and at the first, second, third trimesters, as well as 3 and 9 months postnatal. What you see is evidence for remarkable consistency. That is, in fact, that the evidence for increased levels of depressive symptoms actually pre-exists conception, so that it's evidence in the preconceptual phase. And our study and a number of others have shown considerable consistency over the antenatal and postnatal period in the level of either depressive or especially symptoms of anxiety. But to come to your point, the initial study was a singular assessment antenatally at 26 weeks with follow-up in the postnatal period.
So would that suggest then that the onset of depression is not a critical window of depression related to outcome of the neonates?
Yeah, I think that's really quite true. I mean, there has been, of course, an enormous emphasis on the phenomena of what we consider to be postnatal depression, which implies, although there's very little evidence for it, a unique onset within the postnatal period. Most of those cases are readily apparent in the antenatal period. And in fact, what you commonly see, and there's actually a colleague, Josep Carr, at Nanyang Technical University in Singapore doing ecological monitoring of women over weeks, you see women fluctuating, and what they do is they fluctuate within a particular range. So they may go in and out of the actual clinical range, but they remain within a high subdromal range at very least. And that seems to be highly consistent over the course of antenatal and postnatal periods, at least up to 2 years postnatally.
Thank you.
My pleasure.
Question over here.
Hi, Jason Carmel from Burke and Blythedale in New York. For Dr. Ohls, the effect size of EPO and Darby is really quite amazing. And I'm wondering 2 questions. Number one, have you had any discussions with the FDA about using the multiple phase 2 trials as possible evidence for approval for neuroprotection? And second, have you noticed any change in clinical practice, either at your institution or others, based on these trials?
So to answer your first question about having our studies inform the FDA's decision to add neuroprotection as potential use of ESAs in babies, that's certainly a concern, and we do have an IND for our studies, so the FDA is aware of them. We have to report to them every year. And changing a label is always difficult. Changing a label for a population that is going to use a very small amount of the drug is also difficult. But we certainly have let the FDA know that we would like to have our studies inform the discussion about using ESAs in babies, even just to decrease transfusions, really, but also for the neuroprotective effects. And there are a number of studies right now in place looking at for both term babies and preterm babies and neuroprotective effects. And your second question?
About clinical use.
Clinical use. And I would say that there's definitely been a trend to an increased use of EPO. let's see, I started studying it in 1989, and I came to New Mexico in 95, and I finally convinced my colleagues about 2 years ago to start using it. So it took me about 15 years, 20 years. But there is a trend to it. There was a significant decrease in the use of EPO in preterm infants following a 2006 Cochrane Review analysis that associated EPO with ROP, with worsening ROP, which is why I've mentioned it in all my slides. And that Cochrane Review had some mistakes in it that have been corrected in a 2014 revised edition of it, including the most recent studies. Our study, the study from China, and the study out of Switzerland far exceeds the number of infants that were actually reported in those original Cochrane reviews. And in fact, there's no statistical difference between the placebo group and the EPO groups in the incidence of any stage of ROP. So I'm hopeful that, groups will continue to increase the use of EPO in preterm infants.
We'll take one more question over here.
My name is Naomi, biologist from New York, and my first comment to Dr. Meaney is, wow. Second comment is this being publicized so parents can know? It sounds like with the use of web-based psychotherapy, which has some evidence for cognitive therapy in some conditions, I'm not sure what, there may be services available for people who avail themselves. Also, social services agencies that help parents with low socioeconomic status should be aware that this is an area in need of help also.
I'll return wow, because the last comment, the last portion of your comment is something on which we really, you're absolutely right, we really should be focused. The association between maternal mental health and childhood outcomes, particularly behavioral outcomes, is far greater amongst individuals in low socioeconomic stratum. And common sense just takes you to that place, right? Individuals with greater financial resources have usually a better network within which to operate as far as child rearing is concerned. So you're absolutely spot on. If you wanted to roll this out, I'm editorializing, but if you wanted to roll this out, targeting low SES settings would be an ideal place in which to do so. First of all, as your question implies, you know there's a strong mediational role of maternal emotional well-being in looking at the effects of SES on a broad range of neurodevelopmental outcomes. So that justifies the focus in and of itself. And the fact that what we argue for is a broad consideration of maternal mental health, that those consequences are even greater within that population, argues even further for the point that you're talking about. I mean, if we close our eyes and think of the world, what you'd like to do, particularly if the preconception studies emerge as being valid, is that you'd like to target women in general, and that you'd like to be able to put something up on the web that talks empirically in an evidence-based manner that here's where you are and here's your risk. And here is the web-assisted services that you might access in an evidence-based manner. So that I think is the way, because otherwise we're going to overwhelm whatever health services we have. So I think there's a twofold, just sorry to belabor, but I think you're absolutely right about mobilizing existing services towards impoverished populations and then working to the next level of intervention, which will be, I think, have to be something along the lines of web-based. Yeah, absolutely agree. Thank you.
Thank you.
Dr. Meaney, Dr. Ohls, we just appreciate your presentations. The Academy would like to present you with a certificate and a gift. Thank you. And now it's time for a short break.